Please.
Okay, thank you. Listen, thank you, everyone. Welcome to this great meeting that to the PSP team led by Wendy has put together. I think it's fantastically exciting program. We're going to hear a lot of very novel, as well as updated research that's been going on over the last few years through Prospect and other programs that you've heard on, much of it funded through the PSP Association. I'm very new to this. I just joined the research committee. And I won't say anything more about myself. You've got the biographies of the various speakers and so on. I do encourage people to send in questions. We'll try and safeguard time for questions, because I know from previous exposure and PSP meetings with people affected by PSP, and also, of course, by my other interests, PSP and MND ALS, how vital it is for people to be able to pose these questions and to clarify some of the research issues, which can be complex and often leave more questions than the answer, which of course is good. That's what science is about. It's particularly lovely to hear the message from Michael Coe, of whom I have very fond memories when we were doing various studies in the past. He was always supportive and always asked the hard questions, which were ones that we most needed to answer. And he always, of course, defended the interests and promoted the interests of people affected by PSP. So greetings to you, Michael, from memories or interactions. I'll go ahead now, unless there's anything that Wendy wants to remind me that I should say. I do want to thank the team very much. We'll come back to that and name them at the end. But it has been a work of considerable effort, as you can see from the program, and it's been the fruit of very much work stemming not only from putting the program together, but of course for the development of the research strategy, which I have really had nothing to do with. I'm just as a chairman of the research committee enjoying the fruits. I hope to enjoy the fruits of that, and I hope that it will be extremely productive for people with PSP. So unless there's anything else that Wendy wants me to say, I will go ahead and introduce Hugh Morris. He and I have known each other for many years, but he's really been the preeminent, I think, UK leader of much of PSP clinical programs, including, of course, the great Prospect study. You'll read about his previous work in the biography. And so I'd just like to welcome him and ask him to kick off the first session and with his comments and introduction of Ed Jabbari. So Hugh, thanks very much.
Thanks, Nigel. It's great to be able to join with everyone on this research day and launch of the research strategy. I realised that obviously over the last year it's been a very difficult year for a lot of people, and of course we've you know, I realise a lot of people on the call will have had a lot of difficulties over this year. Obviously, one of the advantages of this is that we've become more used to doing these virtual meetings, which makes it much easier for patients with conditions like PSP and CBD to join in this meeting. So it's great that people can join from all over the country in this meeting today. With respect to COVID, of course, there is light at the end of the tunnel, and you know, in a way, one of the most important messages is that all patients with PSP and CBD should be vaccinated and should take up the opportunity to be vaccinated as soon as that's offered to them. I think it's almost going back 10 years or so that we started to conceive the idea of having a sort of collaborative research network and research study, which became the Prospect study, and it was very generous of the PSP Association to support this. And the aim of this really was to link together research sites across the UK to try and facilitate research into PSP, to drive the ability to have new insights and to develop new treatments. This was coordinated first by John Woodside and now by Alyssa Costantini, who's on the call. I think one of the important things is that it is collaborative, so people are using samples and data and doing analysis of various sites across the UK, and you're going to hear a bit about this morning. At UCL, we've been particularly interested in understanding more about the biology and what insights that might give us into new treatments from a genetic perspective. And again, we've been very lucky to hire Ed Jabbari as our research fellow working on this project, who then has been funded by the MRC to take forward his work on PSP. So it's a great pleasure to hand over to Ed to give us an update on his work on genetics and biology of PSP through the Prospect cohort.
Great. So thanks very much for the introduction, and thanks to the PSP Association for this opportunity to give an update on our recent research developments. So in terms of my background, as Hugh touched on, I was very fortunate to be the Sarah Coe PSP Association Research Fellow for the duration of my PhD in Professor Group at UCL. And having recently completed my PhD, I'm now back in clinical training, but very much remain active in PSP and CBD research. So this is what I'm going to be talking about today. We'll start by talking about the unmet challenges in PSP and how the Prospect study is aiming to address those challenges. Then the main focus is going to be on genetics. So I'll give you a crash course in genetics and genetic studies, and this will hopefully put our recent discoveries in PSP into context. And finally, I'll touch on the current state of play with PSP clinical trials. So a good place to start is a brief summary about PSP and what happens in a brain affected by PSP. And actually, this poster from the PSP Association from 2012 gives a very good summary. And in a minute, you'll see why I've used this particular poster. So we know that up to 10,000 people in the UK have PSP at any one time. And this is a condition that involves a protein that we all have in our brain called tau. And for reasons that we don't fully know, in PSP and also in CBD, tau becomes sticky and clumps together inside brain cells in specific parts of the brain. Subsequently, those brain cells wear away, and only then do symptoms manifest. And around 10 years ago, when this poster was made, the main symptoms of PSP that we were very familiar with were those that were listed in the poster. So things like falls, problems with eye movements, problems with speech, swallowing, etc. So what's changed since then? Well, I want to focus in particular on these 2 points that I've highlighted, which are the symptoms of PSP that we spoke about in the previous slide, and also life expectancy in PSP. Because one of the major developments in the last 10 years is the identification of different forms of PSP, rather than the one classical form characterized by the falls and the problems with eye movements that we discussed earlier. And these alternative forms of PSP have different presenting symptoms, including things like tremor, feet getting stuck when walking, also known as freezing, and also problems with speech and language. And what's very interesting is that a few of these alternative forms of PSP, in particular the ones associated with tremor and freezing, have been shown to have a milder disease course with longer life expectancy in comparison to that classical form of PSP. And the other thing to say is that the identification of these variant forms of PSP means that the condition is likely to be more common than we previously thought. But undoubtedly, and this is a huge source of frustration for everyone involved, there are unmet challenges that remain in PSP. The first is that diagnosing PSP in the early stages is very difficult. And this is mainly because we don't have an accurate objective test like a blood test that can tell us whether or not someone definitely has PSP. So instead, we rely on our clinical assessment of patients, and the challenge is that there is a great deal of overlap in the symptoms and signs of PSP with other neurodegenerative conditions like Parkinson's disease and frontotemporal dementia. So it's not uncommon for patients to be misdiagnosed with these conditions initially. The other unmet challenge, as I'm sure everyone is aware of, is the fact that we still don't have any treatments that can either stop or slow down PSP in its tracks. Although later in my talk, I'll be highlighting the current and future potential avenues for treatments. But what also hasn't changed is the fact that research remains our best hope to tackle these unmet challenges. And the PSP Association and its supporters have and always will have a central role in funding that research. And the Prospect study is an excellent example of PSP Association funded research that has really led to meaningful discoveries, as I'll show in the next few slides. So for those not familiar with Prospect, this is a UK-wide study of patients with atypical Parkinsonian conditions, including PSP and corticobasal syndrome, or CBS. And this study is led by Professor Morris with 7 main UK sites that you can see on the map on the left that follow patients up over 5 years, and a much wider network of over 30 sites that follow patients up over a shorter period of time. Now, in terms of the assessments that patients have as part of the study, these are things like clinical assessments, cognitive tests, blood and spinal fluid tests, and also MRI scans, which Professor Rowe's team lead on at Cambridge. Now, recruitment to the study started in 2015, and we're currently up to a total of around 600 PSP and CBS patients across the UK. And one thing in particular that I'd like to highlight is that remote assessment of patients over the phone has continued throughout the pandemic and lockdown, thanks to the efforts of our study sites and also to our Prospect study coordinator, Alyssa, who many of you will be familiar with. And just over a year ago, we published the baseline analysis of Prospect study data. And you can see from the red circle that I've highlighted that we have a range of patients with these different forms of PSP. And we went on to show that these different forms of PSP have very distinct features, including the symptoms experienced by patients, the pattern of brain shrinkage on MRI, and also the rate at which the disease progresses. So this made us think, well, what are the biological processes that are driving these differences in PSP patients? And what I mean by that is, why does one person develop the classical form of PSP while another person may develop one of these alternative forms of PSP that tend to progress at a slower rate? And you can imagine that the potential benefit of discovering these biological pathways that determine the type of PSP and the speed of progression is that you may be able to target those pathways with medications that may slow down the progression of the disease. And a great way to answer this scientific question is through genetic studies. So that's where we come to our crash course in genetics, which will hopefully wake you all up rather than send you to sleep. And I'm hoping that this will be a simplified explanation that will help to put our recent discoveries into context. So the basic concept is understanding what a gene is and what DNA is. So we have around 20,000 genes in every cell of our body, and each gene contains DNA, which is a blueprint consisting of long segments of these different letters that you can see in the image below. And the specific combination of these letters leads to the production of proteins that allow our bodily systems to function normally. And at each of these letters within the blueprint, broadly speaking, you can have 2 different forms of the letter, and both are completely normal. So for example, at this particular letter that I've highlighted, for example, 70% of the population may have a T and 30% will have an A. Now, while both are normal variants that contribute to the production of a normal protein, genetic studies may find that those who have the A letter have a very small increase or decrease in their risk of developing a particular condition. Now, in contrast, a genetic mutation is where there is a spelling mistake in the DNA blueprint of a particular gene. So again, using our letter T as an example, instead of having a T or an A, this particular person has a C, which is an abnormal spelling mistake and will lead to either no protein being produced, an abnormal version of the protein being produced, or too much protein being produced. And either way, whichever of these consequences occur, the outcome is the same, and that is that the patient will almost certainly develop that particular condition. Now, thankfully, these genetic mutation spelling mistakes are very rare. And in terms of PSP, we identify these spelling mistakes as the cause of the disease in less than 1% of patients. So it is exceptionally rare and mainly found in patients whose symptoms start in their 40s or early 50s, and they usually have a very strong family history of the condition. So instead, we focus more on the overwhelming majority of PSP patients who don't have a genetic mutation, and our research looks into how these common normal genetic variants have an impact on increasing or decreasing not only your risk of developing PSP in the first place, but also your risk of having one form of the disease over another. And in terms of how we discover these important common genetic variants, to put it simply, we compare the DNA of two different groups. So if we want to discover which genes affect your risk of developing PSP, we'll compare the DNA of PSP patients to those of people without any neurological problems. And similarly, if we wanted to discover which genes affect the speed of progression in PSP, we'll compare the DNA of fast progressing patients to that of slow progressing patients. So now that you're all experts in genetics, I'll spend the last few minutes talking about our latest genetic discoveries in PSP. So the first genetic study we did was to compare the DNA of patients with that classical form of PSP that I described earlier to two variant forms of PSP that tend to progress more slowly. And we did this in 600 PSP patients in total, including patients from Prospect, of course. And we found that a gene called TRIM11 played a key role. And what's really interesting about this gene is that it's been shown to have an important role in a waste disposal system in our cells. And this particular waste disposal system has previously been shown to be responsible for the breakdown and recycling of tau protein. So you can imagine that if this system wasn't working properly, there may be more sticky tau accumulating in different parts of the brain, causing PSP. And that's exactly why there are now drug companies that are actively looking into the potential to boost the function of this waste disposal system with a drug to help clear the accumulation of tau. Although I should stress that this is very much in the very early stages of development at the moment. But what has more immediate relevance is our latest genetic finding. So here, instead of comparing the different forms of PSP, we compared the different rates of progression in 1,000 PSP patients, again, including Prospect study patients. And we found that a gene called LRRK2 determines how quickly the disease progresses. Now, what's most exciting about this particular finding and what surprised us greatly is that LRRK2 is a gene that has previously been associated with Parkinson's disease. So this finding suggests that there is a genetic link between PSP and Parkinson's disease. And there are also ongoing clinical trials involving Parkinson's disease patients where drugs are being used to target LRRK2 to hopefully slow down the disease in its tracks. So as you can imagine, our focus now has very much turned to actively work on the potential to set up a similar trial for PSP patients in the near future. But in terms of the current state of play with clinical trials, there is a lot going on at the moment. Again, if we're comparing things to 10 years ago, the amount of interest in PSP from drug companies is much higher these days. And this is largely because of the pathological similarities between PSP and the most common cause of dementia, Alzheimer's disease. And what I mean by pathological similarities is that both conditions involve a buildup of this sticky tau protein. So from 2018 to 2020, there were 2 separate global trials of tau antibodies as potential treatments of both Alzheimer's disease and PSP, respectively. And the way this approach works is that an antibody designed to target tau is injected into a vein peripherally, where it travels up to the brain and sticks to and neutralizes free floating tau that's found in the space between brain cells, as you can see from the image. And therefore that hopefully reduces the spread of tau throughout the brain and hopefully slows down the disease in its tracks. Now, unfortunately, both of these trials did not show a benefit in PSP patients, although a third global trial with a slightly modified version of the tau antibody is due to start later this year, COVID pending, of course. So watch this space. And as Wendy touched upon, Professor Morris will talk a bit more shortly about clinical trials. The other approach that's being explored is tau antisense oligonucleotide, or ASO therapy. So this approach, in simplified terms, is where a drug is given which alters the genetic blueprint within a particular gene, such that it shuts off the production of the abnormal protein that's causing the disease. So this is especially relevant to those very rare cases we spoke about earlier, where there is a spelling mistake mutation in a gene. So in those very rare cases of PSP caused by a genetic mutation in the tau gene, there is an excess of an abnormal version of tau protein being produced. So by giving this ASO therapy, you turn off the production of tau and again, hopefully slow down the disease in its tracks. Currently, large-scale trials using this approach are in early stages of development, and they're likely to initially be restricted to only patients with a mutation in the tau gene, but there is potential for it to be extended to non-mutation carriers in the future. So I'm probably coming to the end of my time, so I'll summarize the main headlines and outline the road ahead. Firstly, the PROSPECT study has shown that different forms of PSP have distinct features and progress at different rates. And there'll be more long-term data from PROSPECT coming soon. And secondly, our genetic studies have highlighted potential important roles of TRIM11 and LRRK2 in modifying the risk of how the disease progresses. And finally, in terms of a potential LRRK2 targeting clinical trial in PSP, I should stress that although we are actively working on this, there are a number of important steps that need to happen before we get there, including confirmatory genetic and biological studies, and importantly, to see if these genes also have a role in the progression of CBD. Lastly, I'd just like to acknowledge all of our fantastic colleagues and collaborators across the UK and internationally, the PSP Association for their ongoing support and funding, and last but certainly not least, all of our inspirational patients and their carers, because without them, none of this would have been possible. Thanks very much for listening, and I'm happy to take questions.
Thanks, Ed. That's a great talk. Wendy, do you want to do questions now, or shall I say a bit more about trials and then do questions after that?
I do a few questions. I think one of the questions was, what is tau? Which perhaps we can just kind of go back to very basics to explain that, you know, tau has been identified as the protein that builds up in the brain. And I really like that analogy you were talking about with TRIM2, about that kind of being the waste disposal kind of truck. You know, if you imagine that the rubbish truck is coming down the road, and you've got all your rubbish building up, and these tau proteins are kind of bags of rubbish, and that you've got this system which is clearing all that away. But obviously, if the truck or the rubbish lorry is broken that day, then that is just going to build up and build up. So I don't know if you could just clarify a little bit more, kind of back to basics, about kind of tau and why we think that is the, you know, important in PSP and in CBD.
So in terms of tau itself, so as I touched upon in the talk, this is a normal protein that we all have in our brains. And you should think of it as a scaffolding protein within brain cells. So it helps the kind of normal functioning of brain cells. And as I said, for reasons that we don't entirely know, tau becomes abnormal in both PSP and CBD. And when those abnormalities occur, that scaffolding function of the tau protein becomes impaired. And also the tau protein itself becomes sticky. And when it becomes sticky, it clumps together. And that clumping together within brain cells leads to those brain cells slowly wearing away and dying. Now, tau protein itself, this abnormal tau protein is not only relevant in PSP, it's also relevant in CBD and also in Alzheimer's disease. And the major differences in those conditions is what parts of the brain that sticky tau protein is building up. And those differences in those brain regions leads to the difference in those symptoms that is experienced in those different conditions.
Okay, thank you very much for clarifying that. One of the other questions is, how many different types of tau are there? Are there different types, or is it subtypes? How does that?
So there's many ways to look at how there are different forms of tau. Broadly speaking, there are two main types of pathological tau that builds up in brains, which are called 3-repeat and 4-repeat. And the 4-repeat tauopathies are the ones that are PSP and CBD, whereas we see more 3-repeat tau in conditions like Alzheimer's disease and frontotemporal dementia. Okay,
thank you. And I guess that also kind of relates a little bit to the different types of PSP, doesn't it? But that's probably another day's kind of discussion. But yeah, and then actually one question actually from one of the other health professionals is around how this links in with the inflammation. And I wonder if I could also invite James Rowe to kind of respond to this as well, if he's there.
Yes, thank you, Wendy, for this time. So we'll just thank you, Ed. It's a fantastic talk, a really clear summary, and it's nice. It's such a good perspective from the basic biology that really is informing trials that are ongoing and others that are imminent. I just wanted to highlight perhaps the importance of those two genes, the TRIM11 and the LRRK2 genes, and they both have a role in inflammation. And I just thought I'd suggest people keep an eye out for that later in the day, as we'll hear this afternoon from Dr. Malpetti about how the inflammation in patients, which we can measure in other ways with PET scanning in live in patients, has such an impact on prognosis. And it's a really nice link between this fundamental genetics and biology of the disease through to prognosis and things that really matter to families in support of trials. So thank you.
Okay, thank you. Yeah, so we'll be hearing a little bit more about how the buildup of these proteins can lead to inflammation in the brain tissue as well. So we'll move on to that a little bit later. On our program, we were planning to have a patient who was going to talk to us about taking part in trials, because one of the things we're going to discuss later is part of our new research strategy is the launch of our Patient and Carer Voices in Research Network, which gives people the opportunity to take part in research, to kind of help in develop projects and look at kind of patient priority research questions as well. Unfortunately, the patient speaker, we were unable to attend, but very kindly, Hugh and Ed have offered to talk a little bit about kind of getting involved in trials that are ongoing and getting involved in trials. And I might be asking them a few questions as well from a patient perspective as how you might do that. So if I can hand over to Hugh, thank you.
Thanks, Wendy. Apologies if I reiterate some of the things that Ed said, but I think it probably does bear repetition. I'm going into a little bit more detail about trials, and obviously, you know, of course, the aim of all the research we do is to actually find something that's going to stop this disease from progressing and actually stop it from getting worse in patients. And that's where things are directed. And an important part of that is drug trials in PSP, which I'm going to focus on. Just to start with, I'll say a bit about COVID-19. So when when the first wave came in March of last year, effectively all of the research studies were stopped from recruiting to on-site for patients, and it was deemed that the risks to patients coming on site would be too great in terms of catching COVID. And also what happened was a lot of the support staff within hospitals who would work on trials like the trials that we were planning and doing on PSP but Alzheimer's disease and motor disease, they were actually redeployed to work on the COVID trials. And obviously the COVID trials were extremely important in terms of discovering that steroids were helpful for COVID and investigating other potential trials for COVID, and actually doing the vaccine trials as well. So actually there was a very big sort of, shifting of the research infrastructure in the UK onto COVID. But unfortunately, of course, that means that conditions like PSP and neurological conditions have kind of missed out, and I think it's fair to say have sort of suffered from what's happened in COVID-19, because there's been a real focus on COVID research and perhaps relatively less emphasis on PSP and neurological conditions over this period. When things sort of returned to normal, we were able to screen and recruit patients to the antibody study that Ed alluded to. But again, of course, we've gone back into another lockdown. So again, new trials and on-site recruitment of patients has been again paused. But hopefully this won't last for very long. I think in a way, one of the sort of benefits of this period has actually been for us to think a bit about how we're recruiting patients. So Alyssa particularly has done a lot of hard work on sort of reconfiguring the PROSPECT study and allowing us to do remote assessments of patients. So to assess people just like this over Zoom, so without patients needing to come up to study sites. And clearly, we knew before that as patients go on, they may find it harder to come up to study sites, but actually having ways of remotely assessing patients is obviously very important for people with PSP. And I think it's a good thing that we've reconfigured our research efforts to enable us to do more research without people necessarily having to travel physically up to a study site to enter into the research study. So it obviously has been difficult from a research perspective, I think, and I know Wendy's been involved in sort of advocating for the research needs of people who've been kind of slightly overlooked or during the COVID-19 outbreak. And I think that's just an important thing we have to focus on next year is sort of building up the research in neurological conditions as well as we sort of all recover from the COVID-19 outbreak. So the PROSPECT network was a research network. So this was the sort of starting sites that were involved in the study. So Chris Kobelecki, Manchester, Alistair Church, Michelle Hugh, Nigel, myself, James, and initially David Burden, then Nicola Pavesi in Newcastle. They have been just tremendously enthusiastic in participating in this, in recruiting patients and carrying out the assessments. And I think that's sort of led to the research that Ed outlined on defining PSP subtypes and the number of people that are likely to be affected with a variance of PSP in the UK, and also the biological research we've done. Obviously, James has led a really important part of the research in this, and Michelle is developing research as well. So there are lots of sort of complementary research areas that are spinning off this research network. We're now up to over 26 sites in the UK and over 900 participants in total, of whom around 600 have PSP and CBD. And I think one of the important things as well about this is sort of...
Can I just, sorry, interrupt you? Would you mind putting your screen in slide view, if possible? I don't know if you're able to. If not, no worries.
Yes,
thank you very much.
I lose a bit of, yeah, okay. So the other thing is that sort of new investigators who have been supported and been very involved with the PSP Association as they've sort of joined to become consultants, have joined in the network. So Boyd Ghosh, who did his research with James, now has a PSP clinic and an active PSP research program in Southampton. So I think it's also sort of nurtured new researchers coming through. And I think in a way, we want to learn more about the biology of this and understand why this condition develops and progresses and how we can develop new treatments. But actually many of the sites that are in the Prospect network have also been involved in drug trials and trying to recruit people to drug trials. So in a way, this has sort of established an infrastructure that has enabled sites to show that they can, you know, do research in PSP and recruit people to drug trials. So I think that's all positive. There are, as Ed alluded to, some disappointments, which I'll recap on. So this is sort of what was alluded to about tau protein. So tau stabilizes these sort of motorways, microtubules that run through nerve cells and carry traffic up and down nerve cells. We think that what happens in PSP is this tau protein becomes sticky. It falls off these microtubules and it forms these abnormal tangled clumps of protein, which clog up nerve cells and also disrupt this trafficking up and down nerve cells. So we think this is at the core of the disease process. And this is sort of an overview of all of the different things, some of which Ed alluded to, and some of which you're going to hear more about later, in terms of the different steps that may be involved in PSP. So we think that there may be an increased production of the protein, so increased expression of the tau gene. The tau protein may be changed in ways which makes it more sticky and aggregates. A very important insight over the last 10 years really is that abnormal tau protein is released from nerve cells and then travels from nerve cell to nerve cell in the brain, and if you like, can infect adjacent nerve cells to cause the spread of disease through the brain. So it propagates, and that's been a real target of therapeutic approaches over the last 5 years. The nerve cell has a waste disposal system which gets rid of abnormal tau protein, and TRIM11 may well be an important part of this. There are also then sort of, if you like, other things that happen. So there's loss of normal function of tau protein, there's inflammation that happens in the bits of the brain that are affected, and there's also problems in the energy supply. And actually many of the areas on this slide have been targets for therapies in PSP, and we hope that targeting one or perhaps several of these mechanisms will lead to new and better treatments for this condition. So sort of going back 5 years, there were 2 antibody treatments for PSP, and the idea of this is that patients would have a monthly infusion into a peripheral vein of antibody. About 1% of that antibody crosses into the brain. The antibody binds to tau protein, and the idea is that when the tau aggregates are sort of floating between nerve cells, that the antibody would bind the tau protein and mop it up, so stop it spreading from nerve cell to nerve cell and causing spread of pathology. So the idea was this would stop the disease spread. So the first, drug trials are quite a laborious process, in that there are lots of steps in the trials before they get licensed as effective treatments. The first step is to see whether the drug is safe and whether it actually does what you think it's doing. So whether it, in the jargon, engages with the target. So there were 2 antibodies that were developed, one by Biogen, one by AbbVie, and there were 2 small-scale studies of these antibodies in PSP with 48 and 32 patients. The results of these studies were that they were safe. They didn't cause side effects. Patients with PSP didn't have adverse effects to peripheral tau antibodies. And in fact, they also were able to show that it bound to tau protein. So this is analysis of spinal fluid from patients with PSP. And perhaps if I direct you to figure 3 here, which my cursor's floating over, this is the level of tau protein in the spinal fluid that's measured with a specific assay. These are patients who have placebo, so this is the normal level of tau protein with no treatment. These are patients who've been given the antibody drug. So this is an antibody drug into the brain. This is now into the vein, sorry, which spreads into the brain through the blood supply to the brain. And this is measuring the tau in the spinal fluid. And there's a big drop in the amount of tau you can measure which implies that tau is being mopped up or that it's being bound to by the antibody. So this is really important because it shows that these antibodies get into the brain and they're able to bind to tau protein in the brain. They kind of do what we expect they're going to be able to do. So this was extremely positive, and we were very optimistic that this was going to be an effective treatment. Unfortunately, the two larger scale studies, so the Passport study recruited 459 patients, and the ARISE study recruited 330 patients. Unfortunately, just about a year ago, we learned the Passport study was negative. So there were no, there was a 2-to-1 recruitment of patients into the active antibody arm and into the placebo arm. This is really important to try and work out whether or not things are effective, that we do have placebo arms in these studies. And these were unfortunately both negative. So there was no difference in the rate of progression of PSP between people who had the active treatment and the placebo treatment. So that was obviously very disappointing. There was a third antibody which Ed alluded to, called UCB0107. So that has recruited to the safety study, which is ongoing in patients with PSP. That study was very disrupted by COVID in that it was very staggered in patients recruiting because of COVID this year. Currently, this is not going to, the results of this study are not known, but currently it's not planned that this is going to progress as a PSP antibody agent. But clearly, you know, I think that this looks like a very promising drug, and we'll need to see what the results of this study, which are ongoing, are. So this is no longer recruiting PSP patients. Ed alluded to and described the second sort of biological treatment. So there was a lot of publicity 3 years ago now for this type of drug treatment for Huntington's disease. So this is called ASO treatment, or antisense oligonucleotide. This is a treatment which turns off the gene expression. So if the gene makes a protein like a tau protein or Huntington protein, that the expression is turned off and the protein levels drop down. So there are a number of trials that have been planned in both Alzheimer's disease and now in PSP. There are 2 ASO therapies that are known to be effective, one for spinal muscular atrophy and one for a type of protein buildup called amyloidosis. So we know in some other conditions that these therapies are effective. These therapies need to be given currently by lumbar puncture, so by spinal infusion. So they're very powerful drug treatments, but they are currently quite invasive in terms of what needs to be done or what patients need to do to participate in these studies. But I think that we can be optimistic about the progress. This is some work from animal studies showing this turning off of the gene. I direct you to this figure C. This shows the progression of loss of brain volume over time. So this is baseline. This is 6 to 9 months, 9 to 12 months, and this shows the atrophy, this shrinkage of brain structure. These are mice that have been treated with this therapy against tau, and actually shows that it brings the brain structure back to normal. So in animal models, which are important, these therapies look promising as therapies for PSP and related conditions. So we think that we're going to be recruiting to a study which is this year, which is this study funded by Novartis. Their drug has got this code number, and we hope we're going to be recruiting PSP patients this year. But currently, this is paused because of the current situation with COVID. I think the other thing to really emphasize is that when I talk to patients about being involved in trials, obviously at the start of the trial, we don't know whether or not the treatment's going to be effective. So, you know, if we knew it was going to be effective, we wouldn't need to do the trial. So there have to be people who go into the placebo arm, and it's extremely generous of patients to volunteer to be involved. And it is a lot of commitment and effort for patients and their carers to be involved in these studies. And of course, the studies wouldn't happen if patients didn't make those commitments. So this is extremely important. The other thing about it, I think, is that being involved in a trial, although of course we really hope that it's going to help the patient, and the patient really hopes that they're going to be helped, ultimately being involved in a trial is altruistic. So the only thing we really know for sure is at the end of trial, we're going to know whether that treatment is effective or not. And I know there may be some people on the call who may have had, may have participated in the passport study, the antibody study, or may have had relatives who participated. And it's obviously very disappointing that that was ineffective, that study. But I think, what is very important is trying to learn the most that we can from the trial. So myself and Ed at the moment are engaged in trying to analyze the trial data with the genetics from the NIP study that Nigel led. So that was a groundbreaking study in terms of a large-scale study of PSP. And although that closed several years ago now, that's something where we can still learn more about the condition, more about how it progresses, more about how it might respond to treatments by reanalyzing the data in the light of the genetics. The same with the Untitled study, which Adam Boxer led, which again, some people may remember, which was ineffective, but we've learned a huge amount about the condition by, reanalyzing the data from these trials. And I think it's really important for, patients and PSP Association, Cure PSP, to continue to represent to drug studies that we'd like to reanalyze the data from these studies to kind of, make the most of what patients have given us in terms of participating in these studies. So in summary, there are biological therapies that are still in development. We've had 2 disappointments in the tau antibody trials so far, but I think there are exciting biological therapies that are in development. I think it's really important that we learn as much as we can from previous trials that have gone on, and there are new trials that are in setup at the moment, which hopefully will start this year. Thanks.
Thank you very much, Hugh, for that summary. That's really, really useful. I have a few questions. One of which is, where are all these trials based? How can I, well, not me personally, but the question is, how can you get involved in these trials? Are they just London based or Cambridge, or are the trials that they can get involved in all over the country?
Yes, obviously it varies from trial to trial, but in general, so the Prospect study does have, which is observational, so that's just collecting information, learning more about the condition, which again, people have been very generous in giving up their time to participate in that. So that does have sites all over the country, so 26 plus sites. And obviously yourselves, the PSP Association, and Alyssa, who is our coordinator, kind of direct people to their local sites or to help people be involved in this. In terms of drug studies, it is variable, but there's been quite a big spread of sites. So certainly Newcastle, Liverpool, Southampton, Cambridge, Newport in Wales, Oxford have all been involved in the drug studies. So there's quite a wide distribution of sites, and I think that's good because obviously we can't. Obviously it's not ideal to have patients traveling long distances to be involved in these studies. So there's a distribution of sites, but it does vary a bit from study to study. And again, I think obviously that we liaise with the PSP Association. I think yourselves having the knowledge of which sites are ongoing with these trials is important. The problem is as well, I would say, with the communication side of it, is that it takes a huge amount of time to get the trial set up. So it goes through the ethics committees, it goes through the R&D departments, and in a way we don't want to, say to people there's going to be a trial next week, because there may be lots of pitfalls, which means it may take 3 or 4 months to get started. And that's very frustrating for patients and very frustrating for for people, research staff that are involved in these studies. So unfortunately, it is a bit of a stop-start process, and even more so with COVID. But obviously, we try to communicate by yourselves where the study sites are for studies that are getting going. Okay,
thank you. Yeah, I think James, did you have something to say about that?
Yes, thank you. Just 2 points. First is that because the PSPA's support for the research network Prospect, the UK is actually internationally one of the prime sites for conducting these trials. So it really is a direct result of this investment from PSPA in recent years, that the international companies look to the UK because it's well organised. And through the network, and therefore through the PSPA, we'll be able to communicate what trials are coming up and where people can look to be recruited. But, and I'm afraid I do have a big sort of note of caution, that the first wave of trials, first time that these drugs are used, there's quite strict what we call criteria or rules as to who can be in and who isn't. It's like the Goldilocks situation. people who are mustn't be too severe, they mustn't be too mild and early either. They've just got to be in that middle ground. And people who've got other health conditions are often not eligible. And our experience is perhaps only no more than a fifth of people with these illnesses, PSP or CBD, are likely to be able to make it into the first trials. This is a source of huge frustration, I think, for us and you. But the hope is then that if we can get those trials to work and succeed, then these treatments will be available to the full spectrum of PSP. But the first trials are really quite restrictive. But do talk to your centre whether you might be eligible.
Yeah, I think that's one of the things we hear as an association, that often that there are all these trials, and either you're not living in the right place, or the condition doesn't meet the criteria. And I think that can be very frustrating for patients, because they really want to, even if it's not necessarily going to be something that might help them, they want to be able to help the next kind of generation of people with these conditions. One of the other questions was, do we have any timeline for kind of drug development, or are we very kind of at the early stages? I don't know who would like to answer that question, but I'll open up to all of you.
I think two or three people might have two or three different opinions, but Hugh, do you want to go first? I might suggest.
Yeah, the problem is that when we get an effective treatment for this, it will be a breakthrough. It will be a real step forward, and it's always difficult to predict when that will be. I must say, this is why trials are so important. I was optimistic about the antibody studies. We know the antibodies get into the brain. We know they bound to tau protein. We know tau protein is spreading between nerve cells. So I was optimistic about those studies, but unfortunately they were negative. So there was no difference between people with the active treatment and placebo. So I think it is very difficult to predict, and it can be a slow process because of all the regulatory aspects. But we've seen with COVID how quickly trials can be mounted, and when people really put a huge effort into it, in terms of study staff devoted to it and resources, frankly, that actually things can move more quickly. So so I think that it's difficult to say. Nigel's probably the most experienced developer of trials among us here. Actually, Nigel, I don't know if you want to comment on this. So,
well, I'm not sure you should ask me, because my track record in this is not very impressive, I'm afraid. As you know, in the ALS field, we have only one drug. Well, we have a licensed drug, but I think the neurologists and sadly the patients would be the first to admit that it isn't a great deal. It leads to if you like, a statistical effect on progression, but it's not something that's subjectively discernible. And it may be that we have to move through that phase. So I think the message is, you just keep trying. You select the best candidates that you can on the basis of current knowledge. And I think what's exciting about PSP and CBS is that you have this protein. Through the efforts of many of you here, you know a lot about this protein, and you're learning more every year about what's going wrong with it, precisely why it's being mishandled, and what might be done about it. So I think you're well set up. I think people should be encouraged by that. I think in the current situation with technology, as you well know, what you could do even 10 years ago in your genetics, you can do, in what might have taken you 6 months can now take you a day in terms of the technology. And so everything is speeding up. And the more a good idea is, the more targets you have. So I think that the breakthrough could come anytime in the next 5 to 10 years. I could be wrong, but I think it's possible. And I would say that with ALS as well. The other question I would ask everyone to think about is, what can we learn from other diseases? Alzheimer's, motor neuron disease, ALS, Parkinson's. We've already learned the crossover with LRRK2 and Parkinson's, which is fascinating. What can we gain from the trials that have been done in these other neurodegenerative conditions? And to ask the question, of course, which everybody is asking, I know, is this going to require a combination of approaches? Because these are complex diseases with complex mechanisms. And really, it may be that we need 2 or 3 different attack vehicles, attack mechanisms, prongs, to get at the disease at different stages, different mechanisms, different molecular mechanisms. And that's, I suppose, what I'm taking on board from my many years of, it has to be said, clinical trials failure in ALS. But that's set against the ground of many of the trials that we did. We were testing drugs. We didn't really know what they did. We didn't have a good target. And at the end of it, we didn't even know whether we'd engaged the target. And I think we've all learned those lessons. Don't do a trial. It's easily said, much more easily said than done. Don't do a trial unless you have a pretty good idea on the rationale, the target, and we all know at the end, even if it failed, did you hit the target? And you've got tau, and you've showed the data that you can show changes in tau with the different strategies. So I think if we build the biomarker and molecular work into our trials, and it costs, and the companies have got to be persuaded to do that, then I think we will move ahead much more rapidly. I'll stop.
Wendy, could I also make a comment? James right here. I think the other thing is, although the trials that we've heard about this morning are really focused on trying to slow the disease down, it's really important we don't forget the need for trials to help the symptoms here and now. And that in fact, if we're lucky enough to have a drug that slows the illness down, the consequences will have more families living with this illness. And it's even more important that we deal with symptoms. We're the UK, where we'll be leading from Cambridge in a matter of weeks. We'll hope we'll get the final regulatory go-ahead for a trial to try and take the edge off the cognitive changes that can be so distressing and disabling in PSP. I won't mention the name of that now, but I'm hoping that through the PSPA we'll be able to announce that really within a couple of weeks. And that I think, because that's not just that particular trial, but the way of focusing on symptoms mustn't be forgotten. It's unlikely that the first trial will be a total cure. If we're lucky, it will slow the illness down, and we then have to have extra ways to help families for living with this illness for even more years than at the moment. Yeah. And perhaps another general point, the headline figures for the recent trials have been of a negative result, of failure, but they've been huge successes in other ways. They've proven the international community can mount these trials one after another, and we've learned a lot about how to run the trials effectively. What do we measure that's important, that's sensitive, so that trial design gets better and better. As people that are listening might imagine, it's easy to show that the drug works. It's actually incredibly difficult to be able to show that even if a drug works, how do we show it? And the trial's getting better and better at homing in on those results and working out perhaps what we call stratification. How do we try and target a drug at the right patient at the right stage of disease? And these previous trials have been extremely successful in giving that information, even though they were disappointing in their headline results. So I think we should actually be quite optimistic as we look for the next few years, although I'm very aware that even a small number of years is a really critical window for everyone listening on this call who's aware of the illness. If we talk about 5 years, that's an age with this illness, but I think there may be movement even sooner than that, I hope.
Okay, thank you. Any other comments, Hugh or Ed? Would you like to add anything particularly to that?
Yeah, I mean, I'd certainly agree with everything that's been said. The only other thing I'll add is one bit of feedback that we receive a lot from our patients is the kind of less inclusion of CBS and CBD patients in clinical trials. And I think that's something that we certainly are working on to improve. That's another unmet challenge that I think we are working on.
There's been a big breakthrough in that, I think, Ed, is that what's held CBS research back for many, many years is the fact that a high proportion of people who look like CBS CBD don't have that biological tau CBS. They have a mimic. Often it's an Alzheimer's mimic. And that's made trials until recently almost impossible to run. But it's become very straightforward recently to, even on the blood test and certainly the PET scan, to separate out the two people, the people with the kind of corticobasal degeneration caused by the true cortical degeneration from those with a mimic, and that I think is going to open the way up for CBD trials. From now, those breakthroughs, particularly the blood test, is just come up in the last few months.
Yeah,
I don't know if you'd agree with that, and it really does make a difference to how we think about a CBD trial.
I've got a couple more questions, if that's okay. So one is about, are there any trials in the Republic of Ireland, and are there any trials in Scotland, or potential, will they be included as potential sites in the future? Anyone able to answer that one?
Yeah, so certainly Tim Lynch in Dublin has been very keen to run trials and has wanted to run trials. In a way, when we're talking about the commercial drug studies, in a way it's slightly outside the hands of investigators, so it's slightly up to the drug companies and where they want to recruit sites. I know that Tim certainly has got a lot of expertise in PSP and has been very keen to run studies. So I think that if anything was potentially available in Ireland, he would be trying to run that. In terms of Scotland, I think Diane is on the call. I don't know if Diane wants to comment on the sort of outlook on Scottish centres.
Diane,
yeah, I'm just unmuting. I'm starting video again. Sorry. I am not aware of active studies recruiting from Scotland at present in PSP and CBD. And I guess I'm buried in an entirely different line. So I hope I'm not misspeaking there. But at present, not that I'm aware of.
I think one of the things we've been talking to about when we have been talking about the pharmaceutical companies is the need for, a potential site in Scotland or Republic of Ireland. We have definitely been kind of pushing for that. So perhaps that's something that we can continue to push for as well going forward from our end, because obviously, all our members are all over the country. In fact, we have members from all over the world, and it would be really, really exciting to get those trials kind of being undertaken further afield. Yeah.
Yeah, I would just say in terms of our recruitment to trials, we get approached by people from other countries about being involved in trials, and actually I've taken the view that we can only really recruit people in the UK. So just because of the NHS system and monitoring side effects and so on, but because I think it is difficult to have. I know that in America they have people flying in and out for trials. I think for ourselves, one of the very important things, the biggest thing with the trial, in my view, when you're sort of starting off, is safety, and just to make sure people are not getting side effects or problems, and actually having the sort of NHS network of local hospitals, GPs, and that back up within the UK, I think makes that, I think that's kind of important. So we've confined ourselves to UK patients for our drug studies. So
yeah.
The other thing to bear in mind is it's a big ask to take part in the trial. They're pretty tough to go through. It's sometimes hard enough to get out of bed or out of the house, but if you've got to travel cross country once a fortnight or once a month for 6 or 12 months, that's a huge undertaking and a commitment for the person with PSP or CBD and their family. So the geography comes to play as well. I think there'd be nothing in principle to stop people from anywhere in the UK taking part in any of the centres. There's a sort of reality about, could you make it? Is it too much to ask to travel 3-4 hours each way to take part? And that could really count against you in the illness. It's so exhausting, can be so exhausting. So
I think I've heard about a day in the life of a research trial. As a patient, it is very, very intensive and exhausting, isn't it? Yeah.
I mean, there are some plus sides that.
Oh, definitely.
Please come in. Just right.
Can I just ask everyone, trialists and others, is there an argument? The key thing, of course, is to get a signal that the new agent that you're using, whatever it is doing something. Quite apart from safety, which is predominant in these early phases, let's just say phase 2 studies, or late phase 1, that is, the first time you move into people with the condition, with PSP or CBD, you need to get a signal of whether it's worth going ahead to a much bigger study, so-called phase 3. So is there an argument from the current data to be lightening the load of all the it takes a whole day, doesn't it, sometimes, to do these assessments? It's a huge burden on the people with PSP and CBD. It's a huge burden on the investigators. The question I always ask myself is, how much is going to be useful out of all this at the end of the day in telling us whether this agent is helpful or not? Because the variance, the statistical variation in many of these tests is huge. You know, it's not easy to devise a good questionnaire. So without reinventing the wheel, how do we get that signal? Now, we need biomarkers, I think we'd all agree. These are molecular tools which tell us what's going on and can tell us whether what we're using is hitting the target and whether it's doing the right thing, what we suppose it to do. Do we need to be a bit more radical in order to make the early stage tests a bit smaller so we can move faster and more informative? And that means not only blood, which we do, but perhaps spinal fluid. We do this in motor neuron disease at the moment. The patients who have been amazing in volunteering and stepping up for this. Now, that may be because we need this anonymity. Maybe with the tau measurements, and this is much more your expertise than mine, we could get away with blood. But do we need to bite the bullet and say, should we be studying spinal fluid as well, in order to shorten the whole process and get a better signal? Because ultimately, that's what we need to move fast for the patients if we can. That's my...
Thank you, Nigel. Yeah, we're actually running out of time a little bit. I just had one other question that I wanted to put to you in this session. There's a couple of other questions actually, which I'll hold back until other sessions because I think they're more relevant there. But alongside all of the, you know, looking at treatments for, you know, the kind of the cure, shall we say, is there anything being done on actual preventing getting it in the first place? Is the question. How do we look at that? Is it possible to look at that?
Yeah, well, we don't know very much about the risk factors for developing this, and there's been kind of interest in sort of environmental toxins, but I think none of them are really clear cut. We know that, you know, obviously the prevalence of PSP is low, so the lifetime risk is low of developing PSP. I think the only thing that, you know, we are learning more and more about in terms of risk factor for tauopathies related to PSP is head injury. And obviously there's a lot of research that's going on into repetitive head injury and the effect of that on people developing tauopathies. So that potentially is something, and obviously various sports bodies reviewing their data on what could be done to prevent potential tau related problems related to repetitive head injury. So that would be a major preventative. That would be a major preventative, avenue to pursue. Beyond that, I'm not sure that we know very much about, you know, preventative strategies to prevent people from developing this, unfortunately.
Yeah, and I think that's a question I often get asked from people from the helpline. You know, I was exposed to this, or this happened to me. You know, what is the evidence that this may have, you know, contributed? And I think it's very difficult to say because we just don't have the data, we don't have the numbers. And I know even for, I know I'm referring to a different condition, but for example, in breast cancer, there's been a study ongoing for 20, 30 years, where, you know, every 3 years you're sent a questionnaire to fill in. And, you know, even that, they've got, you know, that's a huge study, and even that, you know, that's progressing quite slowly. It's quite difficult, isn't it, when there's
Wendy, well, I might better speak to that from the data. So because there's such a low rate of genetic cause of PSP, so there are genetic risks, but there isn't really very commonly a faulty gene that gives somebody in a family PSP, and that's a good thing really, in that it seems to be mostly a one-off in a family. It's not something that gets inherited through the generations. That's a real bonus for people with the condition. but it really makes it more difficult to conduct some of the research because we don't have that genetic handle. And in contrast to other illnesses, like some forms of Alzheimer's and other dementia, which do have a more genetic component, that means you can look 10, 20, 30 years before the illness comes on. It's been very difficult in PSP to know what happens beforehand. And it comes on sometimes so explosively. But everything we know about those other types of dementia is that they begin decades before. And there's no reason to believe that's not the case with PSP. And we've got a study which we're nearly ready to submit, looking at results from what's called the UK Biobank, which had 500,000 healthy people recruited 10 to 15 years ago, typically in their mid-50s, and then followed. And 10 years on, we've identified nearly 150 people who've gone on to develop PSP, who, when they were recruited, appeared, and they believed them to be fully healthy, they appeared healthy 10 years before. But when we look at them closely, they weren't quite normal. So we do now have evidence that PSP was brewing, if you like, quietly in the background, hidden, but beginning a good 10 years before symptoms, just like every other disease.
Yeah.
And that's really important when it comes to some of the concerns about environmental effects or knocks on the head or exposure. People sometimes say, oh, I had an accident and a year later I got my PSP. Well, it's very clear to me that the PSP was beginning well before that knock on the head, 10, 15 years before perhaps. And so it's trying to sort of pick apart what we call bias, which is a sort of artifact of how we remember events. And to really understand that PSP begins biologically very, very early in lifetime, which is both a window to try and treat it before it really gets hold, but also makes it very difficult to study that preventive stage. It's very difficult to find people who are, have PSP, what I might say, PSP in the brain, but no symptoms. And that would be the perfect time to treat, but it's really difficult to find. But we're beginning to understand that, and it does make a difference to how you think about some of these environmental events, particularly knocks on the head or exposure to toxins and so on. They often will come long, long after the PSP has begun, even if it's before the first symptom.
Okay, so yeah, it is very difficult to pin it down to anything specific, but maybe with that data, that will be
exactly. We need to look for that. Yeah.
Okay, thank you. I think it's time for our break. We're a couple of minutes over. Thank you very much for that very impromptu kind of session, which replaced our patient speaker, but I think it was very useful. I do have a few more questions that I'll kind of save for later. If everyone would like to come back at midday, you don't need to log off. You can just leave your screen as it is. It's actually easier that way because you don't have to be readmitted. But if we reconvene at midday, thank you very much. And thank you to the speakers so far. Back, everyone. I'd like to hand over to Nigel, if he's back in, oh, he's not back yet. Just hold on one moment. Oh, there we are. Hand over to Nigel. Thank you.
Okay, thank you. And thanks for our previous speakers and for that excellent session. And it was great to have time for questions. So we now move on to session 2. I'm going to hand over to James Rowe, who's going to chair the various talks. As you know, James is a trustee of the PSP Association and runs the very active research group in Cambridge. And so, James, would you like to do the honours of introducing your speakers for your session?
Yes, thank you. Oh, it's a great pleasure. It's a fantastic day, and I'm really enjoying the session so far with some great work still to come. I'm going to in a moment pass over to Dr. Negin, Holland and Dr. Maura Malpetti. Both have been doing their PhDs in Cambridge at the Cambridge Centre for Parkinson Plus, which is a major investment in PSP and CBD research. I think just worth highlighting that when the centre was founded by the University of Cambridge with a multi-million-pound investment, that grew directly from some seed funding that the PSPA research had given just a few years before, and we were able to magnify that up to what we call leverage, to magnify up additional funding from other sources, but it all stemmed from that initial PSPA investment. It's all part of really showing how the PSPA plays a critical role in PSPA research and leading the way in attracting funders and attracting young scientists into the field, which is perhaps my next point, which is to highlight that the PSPAs and their fellowships, as we saw with Ed, who began his PhD with PSPA support, and in the next two speakers, are both young scientists who are finding the prospects of a transformative treatments and cure for PSP a really exciting area to commit their research careers to. And I think that bodes really well for the future. The first two talks is really a double act by Maura and Negin, who work very closely together, which reminds me again to highlight that PSP research is a collaborative effort. We'll see this in their talks together. We've seen this in the PSPA's funding of the Prospect National Network, and indeed a collaboration between the researchers and families and people with PSP and corticobasal degeneration. So just highlighting that collaborative nature and the vigor of the sort of young generation of scientists coming into research, I will hand over to Dr. Negin Holland. Negin, do you have screen sharing up and running? Very good. So I'll take some questions coming in from the chat as we go, and then perhaps collate those to ask you both some questions at the end of your two talks. Over to you, Negin.
Thank you. Can I just double check that you can see my slides going forward?
Yes, we can. Yeah,
excellent. That's bear with me 1 second, because now I can't go backwards. Here we go. Thank you, James, for the introduction, and thank you to the PSP Association for the invitation to talk. So as James said, my name is Negin Holland. I'm a neurology registrar and also a researcher in James's group. And together with Dr. Malpetti, would like to share some of our research in brain imaging and how it's helping us understand PSP and CBD. We will split our talk into two parts, trying to tackle two events that happen really as part of a larger cascade of events in PSP and CBD. And before we share our research with you, I'd like to focus on this cascade of events that's quite important in trying to target our main aims of improving survival and improving quality of life in PSP and CBD. So improvements in quality of life and survival are the main aims, but to be able to reach that aim, we need to understand what happens upstream. And the simple answer is it's complex. And as Ed has alluded to, there are genetic risk factors, as we've discussed. There may be possible environmental risk factors. But actually downstream of that, then there is a whole cascade of events, a whole layers of complexity, starting with accumulation of this protein tau, inflammation, breakdown in brain communications, which all lead to problems with imbalances in brain chemicals, loss of brain cells, disturbances in the electrical activities of the brain, which in turn all lead to a loss of connections within the brain. This then leads to some of the behavioral symptoms, impulsivity, apathy that we see in our patients, and also problems with physical movements, which then lead to survival being affected and quality of life being affected. The cascade is complex, but actually we can use that to our advantage, because it means that we can, by understanding each of these layers, we can then target our treatments at multiple layers, not just one. And more importantly, we can try and target layers that are closest and directly linked to survival and quality of life, but also understanding what happens upstream. Within our group, we try and understand all of these layers by using brain imaging. One of the techniques we use is brain imaging, be that to look at the structure of the brain, the connections within the brain using MRI imaging, or looking at the electrical activities of the brain using magnetoencephalography, MEG imaging, or using PET imaging to look at some of the molecules, some of the mischievous molecules or mischievous events that happen within the brain, be that inflammation, accumulation of protein tau, or breakdown of the communications between the brain cells. And that's what Maura and I will focus on using PET imaging, in my case, to understand what happens at the level of the brain cells and how they talk to one another. So the units at the core of the communication between brain cells are called synapses. Some of you may have heard of this name, synapses. We have billions of synapses in our brains. And as I said, they're core to the communication of one cell to another, but also one brain region to another brain region. When communications start to fail, we then have problems with thinking, problems with memory, problems with moving and mobility. And oftentimes I hear patients in clinic saying, I know what I want to do, but the messages are just not getting through. And from previous work, or rather, the way we can look at synapses, or the way we could look at synapses previously, was through looking under the microscope of brains of patients who had kindly donated their brains to the Cambridge Brain Bank, where we see a 50 to 60% loss of synapses. But actually now with new technology, we're able to look at these synapses in life. And this is what we've been doing through one of our studies called the Sanders study, which some of you may have heard about. Using this new technology, we've been able to show that there is a widespread loss of synapses. And what do I mean by that? If you focus on this left-hand side panel here, I've shown you 3 columns and 3 rows. The columns look at the brain in different views. So starting from here, let me just change this to a laser pointer. Starting at the top, this is looking from the back, and this is looking from the side. The top row shows you our control participants, or healthy volunteers, who have lots of synapses shown in red. You can see that here. And as we move on to the next row, our participants with corticobasal degeneration, and then subsequently to our participants with PSP, you can see that there is a loss of that redness, the loss of that intensity, and we have more blue and green. So there is a widespread loss of synapses throughout. To put some numbers to this, if you focus on the plot on the right-hand side, we see about 30 to 40% of loss of synapses in our patients with CBD and PSP compared to our healthy volunteers. And that is, on the trajectory of reaching that 50 to 60% that we see at the end of this illness when we look under the microscope. This widespread loss of synapses in CBD and PSP is directly linked and directly related to performance on our memory tests, cognitive tests, whereby low levels of synapses correlate with lower performance on these tests. And in patients who we have low levels of synapses, we have a more severe PSP or CBD. Okay. So that's the story with the communications between brain cells. But how does tau come into this? How does tau cause mischief to the communications between brain cells? You've heard about tau being a normal part of the cells. We all have tau in our brains. It's a scaffolding, as Ed mentioned, it's a scaffolding of all of our brain cells. And it allows cargoes within the brain cells to move up and down. And that movement, that smooth movement, is key to the function of brain cells. If that scaffolding breaks down, or if that scaffolding starts to clump up together, then that movement of essential molecules for the brain cells starts to break down, and that affects the function of the brain cells. It affects the communication between brain cells. So tau, this kind of clumped up tau, can be found within the cell, it can be found outside the cell, it can be found at the synapse. So what is the relationship between that tau and the brain cells and the communications? The simple answer is it's not simple, it's complex. And the reason for that is because the relation between tau and synapses depends on which stage of the disease a patient is in. if you imagine, so look at the plot on the right-hand side, if you imagine an area of the brain where we have lots of synapses, so here is lots of synapses in this brain area, any brain area. The synapses themselves, they can actually promote the spread of tau. They can promote the spread of tau from one region to another, from one cell to another. So actually, in the early disease stages or in brain areas where there are lots of synapses, that actually promotes tau to go a little bit haywire. But tau is toxic to synapses. We know that from our kind of basic science work. So as the disease moves on into the moderate stages and into the late stages or more severe stages, whereas before lots of synapses meant lots of tau, now the tau becomes toxic to synapses, and lots of tau means low synapses and a loss of these communication between brain cells. It's a very complex notion to get to grips with. So let me just put some numbers to it. Okay. Focus on the left-hand side plot. Here I've plotted the patients who came into our study who had both tau PET imaging and synaptic PET imaging. And we're looking at their synapse numbers and how much tau they've accumulated in each region of the brain, shown in different colors. Okay. And what you can see is that in areas of the brain where there are lots of synapses, so take these green areas, for example, or the purple areas, for example, and you have lots of tau. Okay. But have a look at the gray lines. Each gray line is a participant, is a patient participant in our study. And what you can appreciate is that the relationship between what happens to communications and tau building up, in some people is positive, in some people it's flat, and in some people it's actually negative. So there is variability between patients. If we take that relationship, so if we look at the slope of each of these gray lines and say, okay, well, how does that slope change with how severe the patient's illness is? And if we plot that in the right-hand side plot, so each dot here is a patient participant, what you see is that a patient who's in the mild stages of their PSP or CBD has a positive correlation between their tau accumulation and their synapses. Therefore, in their case, lots of synapses means lots of tau. But in patients who are a little bit more severe, so moderate or severe, then that relationship between tau and what happens to synapses starts to become less positive, and in some cases, it becomes negative. This is important for two reasons. One, it means that no one patient is the same as another. One treatment for one patient may not necessarily work for another patient. And we've touched upon this in clinical trial designs and choice of patients and the stage of disease. And the second importance is exactly that, is this, the kind of result, informs the design of clinical trials at the various stages of PSP and CBD. So a bit of a complex notion to get to grips with, but I'm going to change topics. Rather, my colleague Maura is going to change topics. And in the next part of the talk, we're going to focus on a different layer of that cascade, namely inflammation. But what Maura will do at the end of our talk is to marry these two concepts of synaptic loss of communications, with how inflammation affects it. So I'll hand over to Maura, but I will stop sharing because I think she's going to share her own screen.
Thank you, Negin. It's fantastic for picking apart some of the complexity of the illness and how important that is to understand how we might detect how and when a drug might work. The importance of understanding how people with PSP and CBD are very different one from another. We'll save questions for the end. And move straight on to Maura for the second part of the talk.
Thank you, James. And can you see my screen, my slides?
Yes. Yeah.
Thank you also the PSP Association to give us the opportunity to show also this bit of our research. So in this chain of events introduced in part by Negin, today I will introduce also another key player in this framework, which is neuroinflammation. And in particular, I will show you how this relates to tau pathology in PSP and also clinical symptoms. So for neuroinflammation, with terms neuroinflammation, we refer to a complex cascade that involves a lot of mechanisms, but today I will focus just on macroglia activation. So in the brain, we have different cells that, like, some of them have been already introduced by Negin, that are called neurons, but we have also other cells with other functions and other shapes, and between these we have macroglia cells. And macroglia are cells that are represent the brain immune system, and they have different function very important for our brain. However, when they are perturbed or activated, they change shape and also they become dysfunction. And very important also, in addition to changing shape, they change also their chemistry. And because they change their chemistry, we are able to visualize them using PET imaging, molecular imaging scanning technique. And why we are focusing on neuroinflammation? Because neuroinflammation, and in particular activating macroglia, is bad for synapses and neurons. And in particular, there are growing evidence that show how this activating macroglia is particularly dangerous for neurons in neurodegenerative diseases like PSP, but also other conditions like Alzheimer's dementia, for example. So, like, as I mentioned, we can focus this activity macroglia in the brain of our volunteers. And just to show you an example of what we can see with PET imaging with specific tracers for activity macroglia, I reported for you a brain scan, a PET scan with specific tracers for inflammation from one of our volunteers with PSP. And as you can see, like, it's slightly different from the PET imaging that Negin showed for synaptic. In this case, in this rainbow image, the regions that are red and orange show higher inflammation, while colors like between green and blue show that those regions that they have low levels of inflammation. And in PSP, often we can see how this inflammation is high in subcortical regions that are well described in PSP as involved in pathological processes, like, for example, cerebellum, striatum, brainstem. But very importantly, we see also some spots in cortical regions, and in particular, as you can see here, in the frontal lobe. And this is important to mention because in our volunteers with PSP, often we don't see just motor symptoms, but also cognitive changes, behavior changes that are related to these regions in the brain. And then, like, for research, what we do is basically looking at all these scans in each subjects and trying to average in them to find commonalities between different individuals. And then we can plot our results on different templates for brain. As you can see, for example, these regions, subcortical regions like brainstem and cerebellum, can be represented like from this template that remembers a bit a shape of a bird, and we can plot the results to better visualize all specific regions that are involved. So in a numerous study that maybe some of you already heard, we wanted to understand how this activity macroglia in people with PSP is associated with other aspects of the PSP pathology, for example, tau pathology and neuronal loss. And so, like, we tried to see how they are associated using different brain imaging techniques, for example, PET imaging for activity macroglia, PET with other tracers for tau pathology, and the MRI scan for atrophy. And what we found is that in these subcortical regions, we didn't find just high levels of inflammation, but also high tau pathology and tau aggregation, so in the same regions. And that these two processes were related to one another in sense that higher level of inflammations were related to higher tau aggregation in the same regions. But very interestingly, also in the same regions, but a bit more severely and spreadly, we found also atrophy in these subcortical regions. And both inflammation and tau pathology were related to atrophy in the sense that higher inflammation, higher tau aggregation in these regions were related to more severe and aggressive atrophy in these regions. But we went also, we were interested not just how these pathological processes are associated to one another, but also we wanted to understand if inflammation is related in some way to the symptoms that we can see in our volunteers. So we tried to correlate across whole brain the inflammation levels seen with PET imaging with the clinical severity measured with the PSPA rating scale, so which measures symptoms, the severity of symptoms. And as you can see in this graph, every dot is one of our volunteers. And as you can see, these results show how higher inflammation in the brain in these specific regions was related to worse clinical severity at the time of the PET scan. So we found a positive association between higher activity macroglia in subcortical regions with worse symptoms and more severe symptoms. But very importantly, we followed up our volunteers with multiple assessment after a few years after the PET scan. And from these multiple assessment, we tried to model how fast our participants were declining over time. So how every year, how fast the symptoms were getting worse. And modeling this, we extracted this rate of decline. So every year we calculated how much points they lost on this rating scale that indicated that the symptoms and the clinical syndrome was getting worse. And we tried to identify which were the best predictors for this change over time. And again, we identified that macroglia activation in these particular regions was predictive of what was happening in the next few years. So for example, macroglia, like as you can see from the graph, higher level of inflammation at the time of PET was predicting how fast the patients were declining over time in terms of clinical symptoms. And very importantly, in our cohort and our sample, we saw that macroglia activation was a better predictor than, for example, MRI and atrophy measures. So to summarize, like in this chain of event, we show just like, because of time, we just focus on just one part of this chain. And in particular, we show you how tau pathology is relating with inflammation and how these two processes are related to synaptic and dysfunction and cell loss and leading to behavioral changes, cognitive changes, and also faster decline in our participant. However, in James Rowe's group and also the Cambridge Center for Parkinson Plus, other colleagues are focusing on other aspects of this framework. For example, they are studying neurotransmitters systems, how regions are connected to one another, and how we could focus on specific symptoms, like, for example, impulsivity or apathy, and how we could try to find a sort of treatment for these also symptoms. And we are collaborating, as also James mentioned, like we are a team that works all together, not just in Cambridge, but also with other centers in the world to with the same aim. So identifying the specific targets for new treatments, better tools to predict the change, to stratify the patients for clinical trials, for better and early diagnosis, and also to improve quality of life of our volunteers. And I want to thank again the PSP Association for this opportunity, and all our volunteers and their family, and James Rowe for all support and all research that is conducting in PSP, and all our colleagues. And if you have any question, we are here.
Thank you, Maura. Very good. Well, thank you, Maura and Negin. I think we have a few minutes for questions now. If we'll perhaps turn, and Chrystalina, if you get ready to sort of take over the reins in at past. But we have time just a few questions here. Now, Wendy will send questions over to me, but there was one perhaps a very general question, which I might just kick off with first, which is about the difference between PSP and corticobasal degeneration. You'll have noticed in some of the talks, they're talked about separately, a study on PSP here or a study on PSP there. In Negin's talk, looking at synapses, we actually brought these two diseases together. And that's just worth touching on the difference. The way I see it is that sort of in terms of their biology, the two illnesses have very much in common. They both involve this sticky junk protein tau and some of the effects of inflammation and loss of synapses, and some of the genetic factors are very similar. They're not identical under the microscope, but they're very similar. However, when they were first described by neurologists 30, 40 years ago, in their typical form, the PSP on the one hand and corticobasal degeneration look very different in their symptoms. So at face value, they look in people with the illness very different. But underneath it, there's a lot in common. There's also what's become clear is there are some people who don't have typical PSP or typical corticobasal, but get a kind of mixture, a combination of the two. There's not two illnesses, it's the same illness, but with sort of different ways of showing up in symptoms. So they can appear very different, they can appear in overlap, but underneath it all, they have a lot in common. And I think that's why the PSP Association, despite the name, actually has a keen interest to support and to do research into PSP and corticobasal degeneration. I think we're very much aware that sometimes corticobasal degeneration is very much a second cousin to PSP, gets much less attention, less trials and less investment. But I think that that needs to change, and I hope as we go forward that will change, and corticobasal degeneration will become equal with PSP in the attention it rightly receives.
Thank you for explaining that, James. I think that's a question that often comes up, and I think it's quite an important point to note, because we have many people say, well, why do you always, you always talk about PSP, not CBD? And I think it's really important.
Yeah, it's really unfortunate because both of them have such dreadfully long and ugly names. It's very difficult to have a snappy title for progressive supranuclear palsy and corticobasal degeneration. It's just too long. I wish we had a short and snappy name for them both, and then they'd get equal attention. I might just move on with a particular question for Negin, which is in response to one of the people on the line. Could you explain how the changes in the synapses that you've measured with PET relates to symptoms? Why is it that they're so important for the symptoms?
And that's an excellent question. I think I'll just mention one point before kind of directly answering the question, in that one of the things that I didn't show in one of our slides is that this loss of brain communications and synapses happens in areas of the brain where there is no loss of volume. So the numbers of brain cells is the same. It's the communication mechanism that's starting to fail. And I think that's really important in answering the next question of how are they related to cognition. And if you imagine an area of the brain as a county or a district, and the synapses of or the communication between brain cells as the telephone lines in each of in each house in this county. If you start taking one telephone line out, or two or three or four, it doesn't really matter, it doesn't really do much. But pull the plug of the central hub, let's say the BT of that county, and then all of a sudden the communication fails across that county. That county, let's say, is the front of the brain, the bit of the brain that's important in cognition, in putting the brake on our inappropriate behaviors, and kind of telling us what's right, what's wrong, and how to perform complex tasks, for example. If the communication in this bit of the brain isn't working, then you see some of these symptoms of impulsivity, of apathy. If the communication is failing in the bit of the brain that's responsible for movement, for moving quickly or moving, at an appropriate pace, then you see patients having falls or having problems with the dexterity in their arms or their legs. So yes, loss of brain cells can cause symptoms, but actually even before loss of brain cells, even before shrinkage of brain, what happens is that communication starts to fail. And even with that, so when the brain volume itself is exactly the same, but the communications have started failing, symptoms arise. We don't need to wait until the volume of the brain has started to reduce to see these symptoms. They happen quite early on. I hope that's answered the question.
Thank you, Negin. Yes, thank you. I'm very happy for us to take further questions later. A quick question that's just come in before we move on, because it relates to the different types of imaging. So we've been asked, how does a DAT scan differ from a PET scan? That's also a really good question. These are all different types of brain scan. I suppose the difference is the thing that gets injected. I think Maura used the word tracer. Sometimes it's easy to think of this as a dye. It's a chemical that gets injected, that travels around the body, gets into the brain, and it lights up something. And what's really important is what that tracer or the dye is in terms of what it lights up. Different forms of injection might light up synapses or light up these inflamed immune cells. They're very much in research. There's an injection that's in everyday use throughout the UK, which is called a DAT scan. The DAT stands for dopamine transport. It's the way that the brain makes its own natural dopamine that's so critical for movement and balance and some types of thinking. So in a DAT scan, it's an injection of the dye that lights up the brain's natural dopamine cells. And it's a way of telling whether those cells are alive and thriving in number or whether they've been reduced. So these dopamine cells, or the DAT scan that shows them up, is abnormal in PSP and corticobasal, and indeed Parkinson's disease, and another illness called multiple system atrophy. So any of those diseases will have an abnormal DAT scan. But if there's been TIAs or strokes and mini strokes or a drug effect or just normal aging, you wouldn't get this DAT scan showing up abnormal. So the DAT scan is a very useful scan. It is a bit like, the way it works is very much like a PET scan, but what's critical is the thing that the doctors inject on the day.
Okay, thank you very much. I have one question for Maura, if that's okay. Quick one, very quick one. You showed that lovely slide with the all the different colours in the brain, and that was PSP. Can something similar to that be done for CBD then?
Yeah, absolutely. Like in our Nimrod study, we didn't recruit the CBS participants, but mainly is just always the same molecular process. So like we can scan whoever with that PET scan, and the difference between syndromes or pathology, like for example, if you compare PSP with Alzheimer's disease, what changes is level of inflammation in different regions. As Negin said, like the symptoms are related to which regions are involved from the pathology. So also like in inflammation, we see differential regions involved that are related differently to different symptoms. So we can definitely see similar patterns in CBS, but probably involving different regions.
In different parts of the brain. Yeah. Okay. Thank you very much for that.
Thank you, Negin and Maura. I think we'll move on to Chrystalina and Antoniades. She's Professor of Neuroscience in Oxford. I had the great fortune to work with Chrystalina a decade or so ago when she worked briefly in Cambridge, but she's gone on to greater things and is a key part of the Oxford initiatives in investigating causes and pathways to cure and treatment for PSP. And she's going to talk about the OxQUIP study and what it's taught us. So Chrystalina, take it away.
Thank you very much, James. It's a great pleasure to be here. And as you said, I've had the great pleasure of working with you when I was doing my PhD in Cambridge, and it's fantastic to see the collaborative work that is happening and getting updated from our colleagues around the UK. So thank you very much for inviting me today to tell you a little bit about a different type of study, if you like, that we've been doing in Oxford. And I guess it answers partly what, Nigel, you mentioned earlier on in the discussion during the earlier talks about finding an early signal. And the purpose of OxQUIP is very much about the technology aspect, which is very much complementary to what the colleagues have been mentioning up to now. So there have been many biomarker studies in the area of neurodegenerative disorders, and OxQUIP, which is short for Oxford Quantification in Parkinsonism, is a longitudinal study that follows patients with both Parkinson's and PSP. We are following patients very frequently with assessments every 3 months, so it's quite sort of a very frequent follow-up of these patients, with the aim of identifying a signal of progression at a much shorter time scale in order to be able to rapidly screen newly developed drugs. And this is important because only a small minority of drugs, as you've heard earlier being discussed, have already been developed, actually make it successfully through trials to the market. And developing and trialing such drugs consumes a vast amount of resources. And the aim primarily of OxQUIP, as with the other studies that I've heard, is to kill off research into doomed drugs quicker and divert both time and money to the next target. And in OxQUIP, we're measuring neurophysiological and cognitive signals to find physiological correlates over a time scale of 3 to 6 months. We are also focused on ensuring that the baseline diagnosis is as accurate as possible. As we've heard from the other colleagues as well, this is extremely important because it is obviously critical to be able to get the right patients into the right trials to begin with. This has been funded by UCB and been refunded to follow up with remote telemedicine, which I'll say a little bit in a few slides. And we're very grateful for working with industry partners to be able to develop some of these early signals, or at least find if some of this technology is giving us the early signals we had hoped for. There are 3 components to better disease measures: more sensitive tests that are objective and observer dependent, and tests essentially that are quantifiable, I guess, on linear scales. Rather than ordinal scales. What I mean is things that measure things a bit more accurate, if you like, with more sophisticated and accurate statistical analysis. And it's been wonderful because technological advances have enabled us to measure some neurophysiological signals very, very easily. And in our study, OxQUIP, we measure rapid conjugate saccadic eye movements. These are the most common types of eye movements we all make. You're making them at the moment, looking at the screen, listening to me, or doing something else around your house. They are very well studied. We know the physiology. We have a lot of both animal and human models, so we know a lot of what normal eye movements look like. So anything that is abnormal usually is because of disease. And to do these eye movement measurements, we're using portable devices such as the saccadometer. I hope you can see my cursor here. There are a number of different portable devices out in the market, and this is just one of them. There's an elastic band around the forehead of the participant. There are 2 infrared cameras. These are cameras that do not record video, so they're not recording anything from the patient's face. They're just responding to movement of the outside of your eye. Hundreds of trials can be measured within a few minutes, and similarly, we're measuring hand tapping very accurately using touch sensor devices. You can see this down here, just tapping from one sensor to the other and getting a lot of very accurate data on our sensors, and we use a number of different devices as well. We're trying to digitize clinical rating scales, both for Parkinson's and PSP, and we've been using UPDRS along with PSPRS, the 2 clinical rating scales that I'm sure some of the patients in the audience have gone through them with the colleagues, the consultant colleagues. And this is what we call a digitized UPDRS. DRS. So one of our patients here is wearing a small accelerometer. It's called kinesia on the finger here. And when we're asking them to do a clinical assessment, we're also measuring, we're also getting numbers to better understand whether what our clinical feeling is actually correlated to that. We're measuring cognition, which I hope you can see the little video here. So instead of doing pen and paper, we're using different types of technology on simple tablets to be able to see if people can do simple tasks, complex tasks, and so on. And last but not least, we measure gait using a body-worn array of inertial sensors. In simple terms, this is a number of different sensors. For instance, if you see the wrist that I'm wearing, the wristwatch I'm wearing here, it's just sort of sensors of that sort attached around the body, and I'll go through them in just a second. So just to show you, don't worry about understanding what all the data looks like here, just to show you what some of the data that comes out of these devices looks like. Here on the upper left corner, each particular trace is a saccadic eye movement, is this very common type of eye movement that you're all making at the moment to be able to see this here. And also, out of all the sensors that I've shown you in the previous slide, we get a wealth of data information, which is both good and bad in a way, because it's good to have a lot of data, but you have to be very careful in how you interpret and how you analyze this data. So for instance, this is the data from the little accelerometer that goes on your finger. And this down here is some of the data that comes out of all the body sensors that we ask our participants to wear. And this is what the sensors look like, and this is what the data from the sensors look like, especially from the wrist sensor here, from this particular patient. And you can see that the wealth of data, again, that's coming out of this is particularly important in helping us understand whether we do need more technology to help us understand what's happening, or whether, on the other hand, and being devil's advocate here, whether some of this technology might not really be giving us the information that we had hoped for. What you see here is a sort of sensors we've been able to measure accurately. For instance, you see here a 2-minute walk test, a sway test, and a get up from the chair and go test. And on the bottom here, I'm just showing you a few of the data sets that we've been receiving that come out of this middle test here called the sway test. This is where we ask the participant to stand separating their feet with a plank that's used for every single patient, close their eyes and stand there for 30 seconds. Normal, and normal I mean someone who doesn't have a neurological disease such as PSP, is anything that happens inside this gray ellipse, this gray area, so like this person here, who's what we call a healthy control. All these wiggly lines around here is the data from different PSP patients, and it's very interesting to see from the previous colleagues showing how variable some of these results are. So each patient seems to be having a completely different sway form, if you like, especially if they're earlier or later in their disease progression. So let me share with you now some of the results arising from this study. Especially using some of these specialized sensors that I've just been showing you. We investigated a wearable sensor array, so a number of different sensors worn on the body, coupled with what we call machine learning approaches as a means of disease classification. And in simple words, this is just using different sensors on a patient's body and analyzing the data with clever ways of analysis, working very closely with our biomedical engineering colleagues here in Oxford to help us understand whether we're missing some important data and some positive and equally negative results if we use something more complicated, some more technological sensors. And what we found, so first of all, let me just tell you a little bit about the aim of this study. It was primarily to help improve diagnosis. As we've heard, PSP is a condition that can look like Parkinson's for several years, and because PSP is different from Parkinson's, it is important to be able to give people quite an accurate diagnosis as soon as possible, really. It is obviously very critical when developing new treatments and trials, and it has long been thought, and if you look at the literature, there have been a number of studies suggesting that there could be differences in these two conditions. So what we did for this study, we analyzed gait in people who had PSP and also had Parkinson's, using a number of these different sensors and accelerometers on each participant's body. So for this study included 21 participants who had progressive supranuclear palsy, 20 patients who had Parkinson's, and also 39 healthy controls. So usually these are the spouses of some of our participants who didn't have any known neurological or psychiatric disorder. So each person completed a 2-hour protocol every 3 months for 9 visits, so almost 2 years. And for this particular part, we looked at the data we collected from these patients who performed a 2-minute walk, the static sway test that I've shown you previously, and a timed up and go task while wearing this whole array of different sensors. So the sensors that they wore for this particular part of the study were 1 sensor on each arm, on each wrist rather, 1 at the front of your chest, 1 on each feet, and 1 on the lumbar, so the lower back. So there were 6 sensors altogether. This is an example of some of the data available from the gait and sway measurements. And what you can see here is that the parameters derived from each of the tasks, the sway, the gait, the get up and go, were overlaid onto the normal ranges. For instance, if you look on the left-hand side, anything within the gray area is what you expect the normal range if someone doesn't have PSP or if someone doesn't have Parkinson's. What you see here is the left and right of each sensor, 1 for the right wrist, 1 for the left, chest, lower back, and 1 for each foot. And you can see that some of these are abnormal and some of them are within the normal range. For instance, here, our analysis software is subdivided into the lower limb, so the feet, for instance, for us here, or the upper limb, the right and left arms. And also down here, we get very detailed data for the lumbar, so the lower back, from the lower back sensor. The right-hand side, you've seen this on a previous slide where you see the sway. And again, this is a PSP participant who had the disorder for a few years and was progressing a bit more quicker than other patients. Here you can see the parameters derived from the timed up and go tasks. Don't worry too much about the details of what you see in these graphs, but this is essentially to show you how much information and how many details, and this is what I mean by parameters, we can get by using more high level, if you like, sensors of what we've decided to use for part of the OxQUIP study. And again, you get very, very precise measurements of how long a turn takes, for instance, here for an individual to do, how long it takes for an individual to stand from the chair and then sit, and accordingly to sit and stand. And again, these are very, very important because they've given us the power to do a more detailed analysis that otherwise, and in previous, even our own studies, we haven't been able to prove, and we thought we weren't having any positive results really. What we found in brief is that we identified 17 distinct gait parameters that separated the two disorders, progressive supranuclear palsy in Parkinson's disease. Again, the right-hand side is just to show you the heterogeneity and the variability really that we do observe with our PSP patients, and that's why we decided to go for every 3 months. So hoping we'll be able to catch what happens at the 3-month point, 6-month point, 9-month point, and so on. And if you look at the graph on the lower left-hand side, don't worry about the details of this, but just to show you how different the PSP population is looking compared to the PD one and the healthy control. And on this axis here, the y-axis we call it, you can see this is just for one parameter. This is when the person is walking and the duration, how long it takes for this person to turn. Just from that one, you can get enough difference between the different disorders, which for us was of extreme interest. And also, if you want to consider, and that's something that we've been discussing with both colleagues in the UK and internationally, how many sensors does one use? And we've been discussing this with a number of different friends and colleagues around the world, because some people use one sensor, some people use many sensors, we've done both. And what we found, just to draw your attention of the bottom line here that's highlighted in red, if you could see that. Don't worry if you can't, I'll summarize it. If you use a combined array of the 6 sensors I mentioned placed all over your body, essentially getting an overview of what your gait is like and what your movement and your movement control is like, you can get an accuracy of around 90%. We could never achieve this just with one sensor. Now, the news for this is that you don't have to have 6 sensors to get this accuracy. You can have 3 sensors, for instance, one on the wrist, one on the lumbar, the lower back, and one on the leg, to be able to get about 85–86% of accuracy, which again is quite promising, and we couldn't see with previous studies we've done. This is my last slide. Just again, don't worry about all the details here, just to show you how important it is to be able to get very, very detailed data from these sensors. Here we show the key parameters emerging from analyzing PSP, PD, and healthy controls, together with what we call the relative importance as reflected. If you see this column here, you can see hopefully my cursor, the weighting, and how important each of the sensors are if we weigh them against each other. And what you can see here is that the lumbar one, which is the one placed on the lower back, seems to be the winner in a number of different features we have looked at. This is not the whole story, and I'm just summarizing this because of lack of time today, and I'm happy to share the paper, and we have another one that will be coming out soon where we followed up from the cross-sectional study I've just shown you here. But just to sum up for this, the emphasis of this study was the diagnosis of PSP from the much commoner Parkinson's disease. And having a highly accurate and specific diagnostic test, it's necessary to obtain a reliably phenotyped population for trials in PSP, as the colleagues have said in their earlier talks and during the discussions earlier on today. However, it is also important for those running trials in other disorders, such as Parkinson's, to have patients with PSP removed, if you like, from the study population, as they typically have a different progression. Some studies of wearable sensors have used only a single sensors, and other studies have used multiple sensors in the ways that I've just shown you today. A key question is how complex this decision is really needed for optimal performance, really. And we found, as I've just shown you, that the lumbar sensor, the one that sits on the lower back, yields the greatest amount of diagnostically useful information, but also of optimal performance, particularly with regarding to specificity required, sort of additional information from sensors on the limbs, what I've mentioned about having one on the wrist and one on the foot. And what we concluded with is that using a wearable sensor array really around a patient's body with this very clever analysis, what we call the machine learning methods, can accurately discriminate PSP from another neurological disorder, which is closely related, as we know, Parkinson's. Choice of how complex this sensor should be, it's interesting, especially for diagnostic purposes. You need a high specificity, suggesting that you probably need a lot more number of sensors, as we've shown partly with our results, while for subsequent disease tracking, which is equally important, you might find that a simpler system might suffice. And again, we're in the process of finishing another big part of our data analysis for a 2-year longitudinal study. I'm going to leave it here, and I'm going to say that there's a number of people in my team that are working behind the scenes, and as James and his lovely team have mentioned, it's all about collaboration. We have to work together. Without exchanging information and talking to the wonderful colleagues around the world and in the UK, we cannot achieve what we would like to achieve and help you, the patients, at the end of the day. I want to thank UCB for funding us and for continuing to fund us and giving us the opportunity to be able to follow up our study, phase 2 OxQUIP, especially for the PSP side, with telemedicine and home monitoring. So we've started that, and I can't thank enough the PSPA and Wendy for all the fantastic support in advertising our study and getting patients to know a little bit more about our study. We've had a tremendous support from the patients. And of course, the people that are at the heart of all of this, as we all agree, I think, in the area, are the patients yourselves, because without your support and without your tremendous dedication in our studies, we cannot achieve what we hope to achieve sooner than later. Thank you very much for giving me the opportunity to talk today, and I'm very happy to take any questions.
Fantastic. Thank you, Chrystalina. That's a superb talk, and there's been lots of questions pouring in. It's just great to see these sort of user-friendly technologies being brought to bear in the sort of fight against PSP. No matter how smart the behind-the-scenes artificial intelligence work is, the front end, that point of contact with someone with PSP needs to be safe and user-friendly and easy, and that really came across. There's a couple of questions that come in on the study. One is, do you have plans to involve people with corticobasal degeneration in the study? Or maybe they're already involved and you didn't have time to talk about that.
Yes, thank you, James. We haven't involved corticobasal degeneration patients simply because of funding, to be truthfully honest, because when we applied for this, it was for a larger group aiming at Parkinson's, and PSP was the smaller part of OxQUIP. And because it became apparent how important it was to get more PSP research, and from the work I've done with you as well, James, in the past, I've been quite passionate about involving PSP patients. We have been focusing only on PSP. My hope is, and we've been trying to get CBD patients. We haven't got any confirmed yet, but I really hope, and I'm doing the best I can to be able to involve CBD patients in the near future.
Fantastic. And on that, if people have CBD or PSP, how can they get involved? Either with, perhaps we'll start with this study, and then Hugh, you might like to come in more generally about how people get involved with the national network. So for Chrystalina, how could they be involved in this wearables technology research?
Thank you. Wendy has been amazing in helping us advertise our study. So if you want to get in touch with Wendy, then she'll put you in touch with myself or a member of my team, and then we'll take you through the process and make sure, A, that you're eligible, and B, that and you're happy with what the study entails. There's nothing invasive in terms of our study, because, as I said, it's all technology based. But again, we very much, if we get CBD patients, we refer them to your larger national network, which again goes to you and Hugh.
Fantastic.
Yeah,
Hugh. Oh yeah, Wendy or Hugh. Wendy, come on, how do we get involved?
Yes, we'll be touching on this a little bit later as well, as to how people can get in touch with us, and then we can put you in touch with the researchers. So stay tuned later, and we can tell you a little bit more about that. Okay.
Fantastic. Thank you. Chrystalina, a quick question. You talked about a turn and measuring, putting a number on the things that we see in the research, but you talked about a turn. What is a turn in your talk? What did that mean?
Oh, great question. So a turn in terms of the sensors that I've shown, and I'm being a bit conservative in my definition for this, because different sensors I've learned over the last few years define turns in a different way. For us, it's a whole 180 degrees. So it's for someone who is walking along a straight line, and we've got a dedicated corridor at the John Radcliffe Hospital here in Oxford doing this. It's for someone walking along on a straight line and being able to do a whole 180 degrees, including the bits that involve if you've got shuffling gait, for instance, or if you've got problems with falls and might have a mini fall, if you like, during the turn. So once you've completed the 180 degree part, that's what's registered as a turn in terms of the sensor analysis, really. Now, if you look at different sensors and different technology, and that's something we ought to be very careful when we're analyzing our data, you have to make sure that the beginning and the end point on the data analysis part are indeed that 180 degree turn. You could define it as a smaller one, but usually it's that 180 degree turn that you see.
Fantastic. And the emphasis on turns is important. Turning is such a high risk event for having a fall and all that can follow from that. Hence perhaps a particular interest in turning.
Precisely.
Well, Wendy, do we have we're on the hour, but I wonder if I had 5 minutes just to talk about some of the other questions, more broader questions that have come in. Would people be able to wait to have 5 minutes?
I hope so. I think there's been some quite interesting questions. It'd be quite nice to get them raised and
fantastic.
Answered.
Well, let's do that. We'll try and go through. Okay, so one is a question for Hugh, which is really around the role of genetics, not just in the research, but perhaps having genetic testing for people in families where there's PSP. What's the role for it?
Thanks, James. Yeah, well, broadly, as we sort of mentioned, PSP doesn't run in the family. So we've talked a lot about genetic factors, these are things that very slightly increase the risk in the general population, or might sort of modulate the clinical phenotype, so change the way the condition affects people. So these are sort of modifying factors, and although they're very useful and interesting for understanding the biology of the disease, they're not really useful in terms of clinically testing patients. Sometimes we do come across families where lots of people have been affected. That's quite an unusual thing to happen, where there's a family history of PSP, sometimes overlapping with Parkinsonism, or sometimes overlapping with FTD, frontotemporal dementia. And I think in some of those families, it is possible that they have a variant in a gene which is running through the family and causes the condition. And we've mentioned tau protein a lot. There are some very rare variants in the tau gene that can cause something that's very similar to PSP. And obviously, James and his group in Cambridge are very familiar with this, and we're very interested in these conditions as well. So I think it's worthwhile discussing with your doctor if that's something you're worried about, family risk. I think broadly we would reassure people that the family risk is pretty low, but we're pretty interested in families where the very rare families where there are multiple people in the family affected.
Yeah, thank you. And I think that would apply also to corticobasal degeneration as well as PSP. It's the same answer. Perhaps we should touch on drug treatments. There's several questions about drug treatments. Someone was asking about what drug treatments can we use. You know, GPs perhaps aren't well equipped. It seems a bit they sometimes make a stab in the dark about what drug treatments to use. I would say this is very individual, that you really need to think about which of the problems faced by this person in front of me with PSP or CBD that I think a drug is worth trying. So it's not, there's no one drug for everybody. There is a good guide that the PSP Association have written, a guide for PSP and for corticobasal, mainly written with professionals in mind, so GPs and other doctors. But there's no reason, I think, why you as a person with PSP or a member of your family or next of kin couldn't also read that, so you can understand some of the thinking that your doctor has, or to take this as a guide and show to your doctor and say, what about this and what about that? Could we try it? These guides are available online. Carol, correct me if I'm wrong, but they're also just about to be updated. So in the next month or two, we'll have a 2021 update on this general guide to help those professionals, including your doctors, to help you manage your PSP. There's a particular question about levodopa, and it came up in the back of the question about DAT scan. If the DAT scan is abnormal, if the brain lacks dopamine in PSP, as it does in Parkinson's, why are drugs like levodopa, Sinemet or Madapar, the brand names, why are they so ineffective in PSP, whereas they work very well in Parkinson's disease? And I think for me, it highlights that Parkinson's disease and PSP and corticobasal degeneration are very different illnesses. And as in Negin's talk, there's a cascade of events, and in PSP, there's a lot more going on than just dopamine loss. There are other things going wrong, other chemicals that are missing in the brain, also some of those connections are going wrong. And you might imagine that even if you give more dopamine with a drug like levodopa, if those cells can't connect further down the line, then that dopamine can't do its business. It can't work if the cells can't connect. And that comes back to the synapse story in particular. So there's a third question. I might pass back to Professor Nigel Lee. I had a question about the anti-inflammatory drugs.
Well, it was really a question after the lovely talk on neuroinflammation. Of course, the question is how big an impact does that have on the actual disease progression clinically? I think we don't really know the answer to that. We know it's there. But if you had to choose a strategy to attack that part of the pathology, what would you go for in a clinical trial?
Yeah, that's a very good point. So just to recap, we know that the inflammation is there in PSP and corticobasal. It seems to go along with how severe it is, and it seems to predict how quickly an individual person will progress. I think it's important for the story of PSP. And it also makes it a very attractive target to try and switch off that brain inflammation system, that brain immune system, to slow the illness down. One has to be careful that many people on the line will know that people with PSP are very vulnerable to getting infections, chest infections, urine infections, and the like. So one has to be very careful about using a blanket approach to dampen the immune system. So drugs like steroids, for example, that give a sort of blanket effect to dampen the immune system are probably more harm than good. There are some, though, slightly more targeted treatments. There's a study going on at the moment at the Centre for Parkinson Plus with a colleague, Dr. Williams-Gray, not in PSP, but in people who have an aggressive form of Parkinson's disease, to use a drug called azathioprine. So this is a long-standing anti-immune system treatment. It's still quite a blunt tool, and it has to be used carefully. But the thought is that if you know that you have a very aggressive form of the disease, then it might be worth taking the risk as well as the potential benefits with a blunt tool like azathioprine. I don't think we're quite there yet for PSP. We want to understand more about which bits of the immune system and how might you turn off the bad bit of the immune system and leaving the good bits of the immune system working. One has to fight off COVID and chest infections and all these other everyday infections. So it's a work in progress, I think.
Thank you.
A final question, Hugh, I might direct this one to you, which is really about this Golby PSP rating scale and how good is it, and does it predict progression? And how does it vary with phenotypes? Could I get your thoughts on that?
Yeah, I mean, it's obviously Larry Golby's an experienced PSP researcher who basically set up the scale to assess patients coming to his clinic. It's certainly something that seems to, it's a sort of back of the envelope scale, which is sort of based on clinical assessment of patients in the clinic. It seems to track the disease progression pretty well. So it's a 100-point scale. And if you measure PSP patients year on year, you see a change in the scale of about an average of 11 points every year. So it seems to track the progression pretty well. And this similar sort of data has been, achieved in lots of different studies, lots of different sites, lots of different investigators. So it seems to be quite a reliable scale, and it's been used by lots of the ongoing drug studies have used this as their main outcome measure. I think there is a question to be had, how meaningful this is for patients and whether this is really measuring the impact of the condition on patients' lives, and whether scales that really talked about the impact on people's lives would be more meaningful and more important than trial outcome measures. There's also a question of whether this is the most sensitive thing, and whether if we had more sensitive scales, and we've heard, obviously about the quantitative measurement from Chrystalina, and whether actually things that were much more sensitive would enable us to do trials with smaller numbers of patients and see differences in the trajectory of patients with smaller numbers of patients in trials. So I think it's reasonably good, but like all things, I think there's room for improvement in the measurement of PSP. Put it that way.
Fantastic, thank you. That's really, really clear, Hugh. So Larry Golby is a great advocate for PSP research. If I dare to advertise our partners in the US called Cure PSP, they have an event very shortly called Ask the Doctor with Dr. Golby, and they're great events for asking questions and hearing his reply. So we might be able to share that link through the talk later on. I believe it's free to attend. We have our own version here periodically, but his is, I think, next week. But we must make sure we give people time for break and over lunch. So I'm going to draw those questions to a close and thank our speakers this morning. It's been an excellent set of talks, all together, linking together. So thank you to all of our speakers. And I think they and indeed the audience deserve a lunch.
I'll just pass over to Nigel to say a couple of words and then lunchtime.
My only words will be, please have lunch and enjoy it. But thank you to all the speakers. They've really been fabulous talks and have taken us forward. And I'm sure everybody out there has shared that. And the questions have been great too. Thank you. See you later.
Thank you.
Thank you. Time for lunch. We'll rejoin at 1:45, please. Thank you.